All right, chapter 13 is going to focus on meiosis or sexual reproduction. Um, so we are able to distinguish between living organisms in part because they are able to reproduce their own offspring. We can explore this a little bit further by looking at genetics, which is the relationship between heredity and variation. Heredity is being able to transmit traits between generations, and variations are showing the differences that we see in appearance between parents and offspring, as well as offspring and their siblings. So in section 13.1, offsprings get genes from their parents by inheriting chromosomes. We do not get physical traits, we get the DNA, those units of heredity, and they get passed from one generation to the next through gametes, sperms, and eggs. The genes each have specific loci. Individual locations are known as locus on chromosomes, and it's the pairing of the alleles between the two parent chromosomes that determines what gene products are produced. We spent a fair amount of time talking about asexual reproduction with mitosis, where an individual is able to pass genes to its offspring without the fusion of gametes. We saw that with binary fission. You can have asexual reproduction for some organisms that are able to just make identical copies of their offspring. That would be your clones. Sexual reproduction is what we're going to be focusing on this chapter, where parents are able to give rise to offspring that have combinations of genes that are unique based on the genes that the parents contributed. So there you have a couple of an organism that's undergoing asexual reproduction, which would be the hydra and then the redwoods. So fertilization and meiosis are going to occur in sexual life cycles. There's going to be both meiosis and fertilization taking place. Life cycles are the generation to generation sequence of stages that occur for reproductive organisms. This is going to be common to any organism that's able to reproduce sexually. What's going to vary is when they go through the meiosis and when they go through the fertilization, whether they're diploid, whether they are haploid. And so that's what we're going to see as we move through this section. With animals, the only haploid cells that animals have are their gametes. Gametes are produced via meiosis, and they will not go through any additional cell division until they are fertilized. When gametes are fused together, a sperm and an egg, they produce a diploid zygote that then divides via mitosis to continue to make additional diploid cells that will eventually generate a multicellular organism. And so that's what you're seeing in this particular cycle. There you have your haploid gametes. They fuse together, they form the diploid zygote. The diploid zygote continues to grow via mitosis into a diploid multicellular organism. So animals specifically, or we're going to focus on humans here. Somatic cells are all the cells other than your gametes that are diploid. They will have 23 pairs of chromosomes. Um, the chromosome that came from mom and the comparable chromosome that came from dad are known as homologous chromosomes or homologs. They are the same length, same shape, and they carry genes for the same inherited characters. You get the chromosome from, again, each of your parents. When DNA synthesis has taken place, when DNA has been replicated, you will have two copies of mom's chromosome and of dad's chromosome. And when they are together, they are known as sister chromatids. So here you can kind of see a visual, a cell that's undergone the DNA replication process. You've got the sister chromatids, you've got a blue and a red comparable sized chromosome from both the diploid, because it's a diploid cell, you have one from mom and one from dad. And then you can have non-sister chromatids, one from mom and one from dad that are this for the same alleles of information because they're representing the same chromosome. And then you can have homologs when you have a blue and a red one paired together because you have one from your mother and one from your father. Karyotypes are used to display these chromosomes. They're basically ordered pretty much from largest to smallest. And then you've got your centromeres, which are holding the two arms of the chromosome together. And so we tend to do this when you're making a karyotype in metaphase, because that's when they're lined up along the midplate. It's the easiest way to have them be as lengthened as they're going to be. In gametes, we have a haploid number of chromosomes, so that would be 23. You only have one chromosome from mom or dad for each of your 23 chromosomes. If it is an egg, the sex chromosome will be an X chromosome. If it is a sperm, the sex chromosome could be an X or it could be a Y chromosome. So of these 23 chromosomes, you're going to have 22 autosomes. Those would be the ones that do not identify gender, and then you have the one sex chromosome. So fertilization, or when the gametes are going to come together, the fertilized egg is known as a zygote. It's going to have a diploid number of chromosomes because it's gotten one set from the egg and one set from the sperm. The zygote then will reproduce its cells, somatic cells, via mitosis. And at sexual maturity, the reproductive organs in both the female and the male will produce haploid gametes. Um, the We'll talk about this a little bit later on this year if we have time, but there are gametes set aside pretty early on that just have kind of been arrested in terms of their cellular development. And when an organism is at sexual maturity, then they will finalize that process and become full-fledged haploid gametes. The gametes are the only cells that are made in humans by meiosis rather than mitosis. And all the other somatic cells, and after you have the fertilized egg, all the other cells are made via mitosis. So they alternate from diploid zygotes to haploid gametes to maintain that chromosome number so that when fertilization occurs, you're getting one set of chromosomes from the female and one set of chromosomes from the male. And so there you can kind of see that in action. Now we're going to look at plants and some algae. These are different because they do what we call an alternation of generations. They have two multicellular forms, that one is diploid and one is haploid. The diploid organism, known as a sporophyte, is able to make haploid spores via meiosis. Those spores will reproduce via mitosis into a haploid form, a haploid organism, which is called a gametophyte. The gametophyte is able to produce haploid gametes just via mitosis. It's already haploid. It doesn't need to reduce its chromosome number. You have to have haploid gametes fertilize to be able to produce that diploid sporophyte. So your diploid sporophyte can use meiosis to produce spores, and then those spores can grow into gametophytes, haploid multicellular organisms. They can continue to replicate, but then you can have two gametophytes unite together to fertilize to produce a diploid zygote, and then you can repeat your process. So you have two forms of multicellular organisms, the sporophyte and the gametophyte. Only the sporophyte is able to use meiosis to produce spores. Those spores then can be replicated via mitosis into gametes. So with fungi and protists, it's a little bit different because the only diploid stage that you have is with the zygote once the haploid gametes have fertilized. The zygote is able to produce haploid cells via meiosis, and those haploid cells are going to produce your multicellular organism with just one copy of those chromosomes. And then because it only has the one copy of the chromosomes, the adult form is able to produce gametes just via mitosis. So fertilization is the only time when you're going to have your diploid form. You're going to have your zygote. That diploid zygote undergoes meiosis to produce two haploid cells that are able to replicate via mitosis to produce that haploid, either a unicellular or a multicellular organism. So depending on your life cycle type, you can have haploid and diploid cells divide via mitosis, but only the diploid cells are capable of undergoing meiosis because that's when your chromosome number is being reduced. Regardless of which life cycle you're working with, you're going to have chromosomes halved, you're going to have chromosomes doubled, and that's going to help to increase the amount of genetic variation present in your offspring. So focusing on meiosis, this is known as reductional division because you're going to reduce your chromosomes from diploid to haploid. And so it takes place via two sets of cell divisions. You do have to have your chromosomes copied before you can go into meiosis. The two cell divisions will result in four daughter cells being produced, with each daughter cell having half of the chromosomes as its parent. So interphase takes place where you have your chromosomes duplicated, your cells have been growing, they've been joined together at the centromeres, the sister chromatids anyway. Your centrosomes have replicated so that you have two of them. I talked about meiosis being reductional division. That's meiosis 1, where your homologs are actually going to pair up together rather than your sister chromatids. And then they're going to separate so that each new cell is going to get one of the homolog pairs, and then the other cell will get the opposite, so that each cell gets one chromosome 1, one chromosome 2, but it's got a sister chromatid pair of that one chromosome. In meiosis 2, you have equational division, where that's more similar to mitosis, where your sister chromatids split, and now you're going to have four haploid cells with one chromosome of your 22 autosomes and then your sex chromosome. So overview, you've got your interphase, you've got your chromosomes that have duplicated. Meiosis 1, the homologs separate, and meiosis 2, the sister chromatids separate. So both meiosis 1 and meiosis 2 are going to involve the phases we saw with mitosis, prophase, metaphase, anaphase, and telophase, as well as cytokinesis. So here you can see with prophase 1, your homologs are paired together. We're going to talk about the chiasmatas that can form. You still have the spindle taking place, you still have the nuclear envelope fragmenting, but your homologous chromosomes pair up together, and then you have them line up in metaphase along the midplate with the centromeres connected to the kinetochores. They get pulled apart by those kinetochore microtubules so that the sister chromatids stay together, but the homologs separate from one another. And then you are going to have a furrow form. You are not necessarily going to have that nuclear membrane completely reform because, again, these cells are going to go through meiosis 2 or equational division. You may notice that there is a little bit of a difference between the chromosomes we started with in prophase 1 and the chromosomes we ended with in telophase 1. And that's because crossing over can take place where the homologs are paired up together at those chiasmatas. And we'll talk a little bit more about that. So prophase 1 is what's going to take the majority of the time for meiosis to take place. You need to get your chromosomes to condense. And then you're going to have, again, those chiasmatas form where the crossing over can take place. Synapsis is the process by which the homolog chromosomes are able to line up together. So mom's set of chromosomes and dad's set of chromosomes line up by genes. And then you're going to have crossing over occur when you have the exchange of DNA between those homolog pairs. They basically are forming a tetrad because you have two pairs of sister chromatids that are paired up together. So you have two sets of homologous chromosomes with their identical copies next to one another. As I said, metaphase, the tetrads line up. You're going to have a homologous chromosome facing each of the two poles. You're going to have the kinetochore microtubules grab on to the kinetochores attached to those homologous chromosomes. And then you're going to have one attached to each homologous chromosome from each of the poles, each of the centrosomes that is at the opposite ends of the cell. An analog, they separate. The chromosomes are going to move towards the poles via the spindle apparatus. The sister chromatids remain intact. Only the homologs separate. The sister chromatids stay together. In telophase 1, you have haploid sets of chromosomes, but you have two copies of that chromosome. Now, they may not be identical anymore, but you have a chromosome that consists of two sister chromatids present in each pole of the cell. So cytokinesis is going to divide the cells into haploid daughter cells. You'll have the cleavage for the cell plate form. There's no need for chromosome replication to occur because you've already got your duplicated chromosomes present. So meiosis 2 again is going to look a whole lot like meiosis or mitosis. Prophase, you've got your chromosomes. They're already condensed. You're going to have them line up on the midplate. You're going to have the kinetochore microtubules join on again. They're going to split the kinetochores at the sister chromatids so that you have sister chromatids from each chromosome moving to opposite ends of the cell. And then in telophase 2 in cytokinesis, you're going to have that nuclear membrane form. You're going to have the cells split, and you truly will have haploid number of chromosomes present in each of the two daughter cells. So that's what we're talking about here with prophase 2. Again, they're no longer identical anymore because they've had the crossing over take place. Yeah, and so each of these daughter cells is no longer identical to any of the other daughter cells due to that crossing over, due to the synapsis process that led to crossing over. So mitosis is intending to replicate things identically. In terms of your chromosomes, you should have the same number of chromosome sets, the same sequence of DNA. Meiosis does not attempt to keep the chromosomes identical genetically speaking. They're going to have the crossing over taking place so that some of the alleles have been exchanged, and the chromosome number will be reduced from being diploid to haploid. And so there's just a visual of what's happened with mitosis and how that would fit in with meiosis 1. Again, we don't see a whole lot of similarities with mitosis until you get to meiosis 2, where you get down to the individual chromosomes. But you will notice with mitosis, you start with diploid, you end with diploid. With meiosis 1, you start with diploid, and meiosis 2, you end with haploid. So both of them replicate the DNA prior to entering mitosis or meiosis 1. Mitosis has one division, meiosis has two. Mitosis does not have any synapses take place. Meiosis 1 has that take place during prophase 1, which is going to have the crossing over due to the chiasmata, which are holding the pairs of homologs together. You're going to produce two identical diploid cells in mitosis. In meiosis, you're going to produce four haploid cells that are going to be genetically different. Mitosis, these cells are going to be able to use to produce your multicellular organism. In meiosis, when you go from diploid to haploid, you're producing your gametes. This is also providing ways to increase the variability in the alleles, which is going to have a pretty significant impact on the genetics for that potential offspring. So things that are unique to meiosis are all basically going to occur in meiosis 1, the synapsis and the crossing over. In metaphase, having the paired homologs split apart rather than having the sister chromatids split apart, and then anaphase, that actual splitting. So metaphase are lining up on the midplate, anaphase are splitting up. All right, and so there's just showing you that visual of what's happening that's different from mitosis. So why we're able to keep the sister chromatids together, they have those cohesions. We talked a little bit about them in mitosis that will help them stay together within an individual chromosome. In mitosis, those cohesions are split in metaphase, but in meiosis, they do not get split until anaphase 1. And then they get split along the chromosome arms in anaphase 1, and they get split at the centromeres in anaphase 2. So the cohesions are going to be throughout the chromosome in meiosis. In mitosis, they are primarily just at those centromeres. So variation that we are able to get genetics wise via these sexual life cycles is going to play a role with evolution. Mutations were the original source of genetic diversity in organisms, which can lead to different forms of genes known as alleles. And when these alleles get reshuffled, we get different combinations of protein products. Factors that can play a role in leading to genetic variation. The fact that the chromosomes can absort independently. There's no particular reason why mom's chromosome 1 has to follow mom's chromosome 2. It could be any particular combination. We have the crossing over in prophase 1, and then just the random chromosomes that are going to be combined together as a part of fertilization. So speaking of independent assortment, again, the homologs are going to orient randomly. So the mother's chromosomes and the father's chromosomes are going to separate independently. And because if we look at it from a human perspective, we have 23 chromosomes. That basically provides for 2 to the 23rd possible combinations of chromosomes that could be separated into gametes. And that's like over 8 million. So there's a lot of possibility for variation just due to that independent assortment. All right, and so there you can kind of see some possibilities. Sometimes you could have both of moms go together or both of dads, and sometimes you could have one of each. No rhyme or reason. Crossing over where we have the pieces of the chromosomes basically switch. They trade places so that alleles that were with mom's chromosome initially are now with dad's. Due to them lining up by genes and having the synapses take place, that too can add to genetic variation with the recombinant chromosomes that are formed. And then fertilization, because you can have any sperm fused with any ovum. They each have over 8 million different possible chromosome combinations. So you put them together with a zygote, you can have 70 trillion different diploid combinations. So more crossing over, more variation, which is why it is practically impossible for you to look identically like um, one of your parents only, or unless you were born, unless you have a twin or a triplet and you were from the same individual fertilized egg, you are highly unlikely to look like one of your offspring or one of your siblings, excuse me. And so the last piece we're going to talk about is how this can play a role within populations. We're going to spend more on time on evolution, probably February or so. Natural selection is going to result in lots of different genetic variations. That are going to be favored by the environment. And so you have all these different allele combinations that are available. And so this is going to increase the amount of variation in a population. And natural selection is going to again, due to that environment, due to that population's desire to survive and be able to reproduce in that environment, the amount of variation you have is going to increase the likelihood of that organism being successful in such an environment with having all the different allele combinations that are possible. When you start to reduce that variation and minimize the different possible combinations of gene products, that is going to make it a more challenging possibility for an organism to survive, depending on what type of environment they're living in.